Welcome to the fourth and final video for our module on evolution. Here we're going to talk about natural selection in detail and talk about all of the processes that are important for uh, evolution to happen. So basically the processes that drive evolution. Um, and so to get started, remember in the second video, I believe, or one of the earlier videos, we talked about how Darwin brought about the idea that species change over time through descent with modification, and that modification came from the selection process or these selection pressures, um, which he deemed natural selection. And one of the key things with natural selection, and I mentioned this before, is that natural selection will act on individual organisms, right? So it will say if an individual mouse has a white coat color on the beach, natural selection will favor that organism versus one that doesn't camouflage with its coat color on the beach. So that individual organism will have a increased fitness value. But the overall process of evolution does not happen at the individual level. It happens at the population level. Okay. So again, remember I said this earlier, the important idea for evolution is that individuals do not evolve, populations do. And the reason it's like this is because that individual organism has a specific trait that it inherited from its parent. Now that trait has a fitness level to it. And based on that fitness level, it will have an increase or decrease survivability rate and reproductive rate. More fit organisms should have an increased survival and reproductive rate. So you would expect that gene to be passed on more often. So if you have two genes, one for light coat color and one for dark coat color, and they both start at 0.5 frequency in the population, so 50-50. If the light is more fit, then you would expect more individuals with this gene to survive and pass it on than the dark. And so you would expect light to increase in frequency and dark to decrease in frequency. And so over time, the proportion of the population that has that light coat color will increase. The portion of the population that has that dark coat color will decrease. So in the beginning, when you had 50-50 light and dark coat colors, that will be very different from the end where maybe it's 100% or like 95% light versus dark coat colors. And so that population evolved over time to express light coat colors more often than dark coat colors when they live on that beach sandy environment. So the individual is responsible for housing and passing on those traits, but the population evolves because the frequency of those traits change in the population over time. And that's basically what the definition of evolution is. Evolution is referring to that generation to generation change in populations. And more specifically, it's the allele frequency that changes. So again, initially we had a frequency of 0.5 for light and dark, and by the end, let's say we were at 0.95 and 0.05. 0.95 was light, 0.05 was dark. So you had an evolution of that population in favor for the light coat color. Okay, and so that's important to understand why individuals don't evolve and populations do, and that's what we mean whenever we reference that. Okay. And because Darwin hypothesized that species basically change over these long periods of time, he knew that he wouldn't be able to specifically study evolution through direct observations, because a lot of the organisms have long lifespans. It takes a long time to accumulate mutations and changes throughout generations, and so it would take hundreds to thousands of generations for us to see these changes. And a lot of times that doesn't happen in our lifetime unless we're looking at fast growing organisms like bacteria, right? And so what Darwin did is he thought about how he could express this idea of evolution, even though we weren't able to observe it today. And so he thought about artificial selection and how humans have been selectively breeding and domesticating plants and animals for thousands of years. And he was like, okay, if this is basically related to the idea of natural selection, but instead of natural selective forces from the environment being put on an organism, humans are the selective pressure, and we are specifically selecting for specific traits that we want in our plants and our animals that we're domesticating, right? And so by selecting for those specific traits and only breeding organisms that have those traits, we're essentially producing a natural selection filter on the organisms, but doing it artificially. And so what he learned through artificial selection is that there are two basic components. One is that there has to be variation within a population. If there is no variation, then there is no fitness differences between individuals. If all the mice had white coat color, then there are no fitness differences. But because you have mice that are light coat color and dark coat color, there is variation, and that infers a fitness value. The second part is that those traits have to be heritable. So you have to be able to pass on a trait from parent to offspring. If you can't pass that trait on, then selection doesn't act on that trait. Okay, so those are the two big important factors for natural selection to happen, and he learned that through analyzing the process of artificial selection. So you have to have variation, you have to have heritability, and we can add another factor to this, a third factor. And that third component is there has to be fitness differences in those traits. So for example, one trait has to be more fit than another. If they're equal fitness, then there's not going to be any selection for either one of those. And so that trait's not going to be selected for. Okay. And so Darwin knew, based off of all of his observations that he's made throughout his lifetime and throughout his research and travels, that natural populations do have small measurable differences. Okay? But one thing he didn't really know is what forces determined which individuals became the quote unquote breeding stock for the next generation. With artificial selection, humans go in and specifically say, okay, we want the strongest horse to breed so that we can have stronger breeds to pull our plows. We want the corn that produces the most yield with the biggest ears, and we're only going to breed those. And so that's how we chose our breeding stock for this artificial selection process. What he was unsure of is how this happens in the natural environment. So how nature decides what's fit versus what's not fit. And so here's a population of Asian lady beetles where you can see a considerable amount of variation in not only coloration, so completely red versus completely black with red spots or black spots or reddish orange, but also variation in the size of those spots and the number of those spots, whether it's on the completely black shell or the completely yellow orange shell. So lots of variation in this population of lady beetles. Now, at one time, there was a researcher, Thomas Malthus, and he basically said that human disease, famine, and war were consequences of human population increasing faster than resources, right? And so the reason humans had disease, the reason we had famine, was because our populations were getting larger than our resources were able to keep that population. So after learning kind of this thought process from Malthus, Darwin applied this same idea to his plants and animals, basically saying that resources in an environment are limited, just like resources for human populations are limited. And that if you have more individuals than what those resources can take, then there's this struggle for existence. And so now only the more fit individuals are going to be able to survive and reproduce because they'll be the ones that can gather the resources and can find mates. And this is because your resources, the number of mates, the number of territories, size of territories is limited. And so the more fit individuals will gain access to those, and whatever traits lead those individuals to have that increased fitness will be passed on. And so only some offspring are surviving each generation, and those are the ones that have those fit traits as well. And so that's going to again select for specific traits to propagate and increase in frequency in a population over time. And so what we see in species is that a lot of times you get species that will overproduce for offspring. So basically they'll produce hundreds of thousands of eggs because only a small percentage of their offspring will survive. Or you'll see species putting a lot of energy in parental care to make sure their one or two offspring survive. And so there's very different traits, very different life history strategies that make sure that species use to ensure that their offspring are going to survive and that their genes will be passed on to the next generation. So in the process of natural selection, again, the individuals who have the more fit traits, which allow them to find resources, to keep their territories, to escape predators, will be the ones that survive and reproduce. And that's the selection pressures, that selection filtering process of natural selection. And so what Darwin basically said is that if humans can take artificial selective breeding, artificial breeding mechanisms, and cause significant change in short periods of time to where we get all of the dog breeds that we have today, then why can't natural selection modify species over hundreds to thousands and millions of generations, right? And so this is the idea that Darwin argued in his book and used artificial selection to point out just how natural selection can work and does work on natural populations. And through research after Darwin, scientists have went in and documented evolutionary change in thousands of scientific studies. And one really common example of natural selection is the evolution of pesticide resistance in insect species. And what we see with this pesticide resistance is that as you apply a pesticide to an insect population, most of the individuals will die from the pesticide, but you'll have a few individuals that will be resistant. They will survive, they will reproduce, and they'll pass on that resistance. So now you have an increased proportion of the population that's resistant. You spray the pesticide again, most of them will still die, but a higher percentage will survive this time. And then they'll pass on, and the offspring now have a higher percentage of resistance. And that keeps happening until most of the population is now resistant, and you now have a population that resists the pesticide that you were using to control that population. And so we'll see the process of selection to amplify and diminish those traits that are heritable. And it's only the heritable traits, it's only the traits that can be passed on from parent to offspring. And so even though organisms can acquire characteristics that help it survive in its lifetime, if they are not tied to their genetics and inheritable, then they're not passed on and not influenced by the process of natural selection. And so another important fact we need to come to grips with is that natural selection is not goal oriented, right? So it doesn't lead to perfectly adapted organisms. And the organisms don't experience a situation and say, oh, you know, like insects, oh, this insecticide is being applied to our environment. We need to develop a resistance. And so they develop a resistance. That trait has to already be in the population in order for it to be acted on by natural selection. So organisms don't develop traits because of a need to have it. That trait is either there and selected for, or it's not. And the reason we say that natural selection does not lead to this perfectly adapted organism is because a trait that's favorable in one situation can be useless in another. And so let's go back to that coat color example. A mouse that lives on a beach that has light color fur is really fit for that environment because he's camouflaged. However, as soon as they go into the forest, they're no longer camouflaged and they're not fit for that environment. And so where they were doing really well in the beach environment, they do really bad in the forest environment. And so again, fitness is determined by the environmental factors that are present in their current environment, right? And so again, it's not forward looking, it's not goal directed. Natural selection only acts on the traits that are present in the population now and is based on the environmental characteristics of that population. And so here's just an example figure of that pesticide resistant. So you have your population of insects. The red is the resistant, the green is the non-resistant. You apply the pesticide, your greens die. Your red is left, they reproduce. Your survivors now the resistant allele. And so now they're able to resist that pesticide and not die whenever that pesticide is applied. And you end up with a resistant population. So now you have to develop a new pesticide or a new way of handling those insects. Okay. So Darwin obviously didn't know everything about evolution, and there were a lot of things he wasn't able to explain. But what was interesting with Darwin is he was intelligent enough and he's smart enough to kind of hint at how some of these things may be happening and occurring. And so some of the things that Darwin didn't know is how the variations bring up or come about in populations. So how do you get the variation in populations? He knew this variation existed, but he wasn't exactly sure how you get new variation. He also wasn't sure how variation was passed from parent to offspring, but he knew that there had to be some mechanism that was allowing that to happen. But at the same time, Gregor Mendel had actually already answered these questions. It's just that Darwin never read his research. And Gregor Mendel's research actually was kind of lost away for a little bit. And it wasn't until the modern synthesis, which was in like the early 1900s, that led to the combination of Gregor Mendel's work and Darwin's work to really bring about these ideas and help us understand more about evolutionary theory. And so now what we're going to get into is where that source of genetic variation comes from that natural selection acts on. So like we mentioned, most populations have variation between those species. You have those obvious physical differences, lots of phenotypic, so the way they look, variation, And we can observe that variation at the molecular level, so at the genetic level. But again, not all variation is heritable. So natural selection is really only acting on that variation that is heritable in that population. So just like with the ladybugs, here's variation in snail population, where you have different colors, different types of stripes, numbers of stripes, width of stripes, etc. So same species of snail, just different variation in that population. And so now how does this variation come about? Primarily it comes from mutation. Mutation is the ultimate source of genetic variation. It serves as the raw material for evolution and serves as that variation source. New alleles will originate by mutation, so just a change in the nucleotide sequence. So let's say you have a sequence that is made up of these base pairs. So let's say you have A, G, G, C, A, right? And this codes for white coat color. Now through DNA replication and synthesis, an error happens. So instead of having A, G, G, C, A, maybe we get T, A. So we get a mutation here. Now instead of being light coat color, maybe now it's dark coat color. And so one mutation like this doesn't always lead to phenotypic changes and variation. Most of the time it can take multiple mutations to do that, but every now and then it does. So for example, sickle cell anemia is created by one single mutation. You change one base in that gene for hemoglobin shape in red blood cells, and you get sickle cell anemia. And a lot of the times these mutations actually don't affect anything, and it's called a neutral change, where it doesn't change the amino acid or the way that organisms look. And then most of the time, besides the neutral, they're usually deleterious. So they're really bad changes to the protein that causes the organisms not to survive. Every now and then, that mutation will lead to a different phenotype that can be selected on through natural selection. And it's also important to note that these mutations have to happen in the gametes, so in the sperm or the egg, because that's the DNA that's being transferred to the offspring. The DNA in our skin cells or our liver cells is not being transferred to our offspring. So if we get a mutation in our liver cell, that's not going to be transported to our gamete, because our gametes will have the original DNA that's not mutated. And so that mutation has to happen in the DNA that's in the gametes that leads to the development of the gametes. And like I mentioned, that single nucleotide can lead to sickle cell disease. Okay. And like I also mentioned, most of the time that mutation will mostly be harmful or neutral, so no change. And on rare occasions, it can lead to a beneficial allele that is selected for will be an evolutionary adaptation. It will increase survival rate and reproductive success. We get these kind of effects more often when the environment's changing in a way to where that mutation used to be disadvantaged, used to be deleterious, but is now favorable. So for example, if like dark coat color mice on the beach, and then all of a sudden the beach becomes forested because of climate change, then that dark coat color is now beneficial. And so you get selection for it instead of for the white coat color. And with these mutations, they actually delete, disrupt, or rearrange the genes. So like we talked about in the previous module, where you can get frame shifts or you can get deletions, and so all of that affects the proteins and the amino acid sequences. And the duplication of a gene in meiosis also can produce genetic variation. And it can also lead to new genes with different functions, and that's through duplication events. So basically, if you end up with an error in meiosis and say you get an extra chromosome that was duplicated, so now you have two copies of a chromosome. One is normal and still functions, but if you get a mutation, so let's say, you know, this area mutates, you still have a normal one, which does the normal function, right? But then this one's mutated and it can do an abnormal function, right? And that abnormal function may be good, it may be bad. If it's good, it'll be selected for and could eventually develop into a new gene, but we're still keeping that normal function, and that can lead to a lot of new evolutionary adaptations. And there are a group of genes that have been duplicated all throughout evolutionary history of organisms that are called the Hox gene. And the Hox gene is important for body segmentation. And so the expression of that Hox gene with the duplications has led to the variety of body plans that we see throughout the evolution of animals and insects and organisms. And so it's a very interesting gene. If you're interested in it, I really recommend looking up Hox genes. Super interesting. Whenever you have organisms that have fast generation times, like prokaryotes, so say bacteria, mutations can really generate and accumulate very quickly in a population, especially when they're haploid. So for example, bacteria are haploid. That means they have one allele for each gene, and that allele comes into effect immediately. It's not like with a diploid organism where a dominant can override a recessive, and if it's heterozygous, you don't see the expression of that dominant, of that recessive allele, and so it can take a little while until you get homorecessive genotypes to show that expression. Right? With haploid organisms, it's immediately expressed, right? Because there's no dominant recessive or codominant or intermediate dominant relationships. And those bacteria have quick generation times. So within a day, you put bacteria on a petri dish and you can get thousands of generations. However, with animals and plants, those generation times are very long, and you average about one mutation in every 100,000 genes per generation. And so that's very few compared to what you get with prokaryotes within a time, the same time span. And so those long time spans between generations and also having the diploid genomes prevent a lot of those mutations from affecting variation and actually showing up in the population. And so that's why Darwin had, he didn't know this detail, but he knew that because of long generation times, he most likely wouldn't be able to see evolution happen and be able to observe it. Now, there are other kind of selection pressures that can affect populations. And so sexual selection is kind of a subcategory of natural selection, and it's where certain traits are favored within the organism and selected for in the reproduction process. And so organisms that reproduce sexually, most of that genetic variation results from a unique combination of alleles that each individual inherits, right? And so you can get new assortments of alleles arising. And remember, we talked about this through mitosis and meiosis. So you get independent orientation, random fertilization, and crossing over. So this is that independent assortment. So those are the three ways you can get increased variation or changes in your genotypes without leading to changes in the genes itself, so mutations. Okay, crossing over does change the genes, but it's not a source of, say, like a mutation. Um, and so with this sexual reproduction process, you have traits that are being selected for, based off of sexual selection preferences. So like big horns or beautiful feathers or amazing dances that the birds may put on. And so those traits will pass on that lead to those behaviors or phenotypes because that's what they're selected for. And whenever you look at populations, right, you can see the impact of natural selection in that population. So remember, it's not on the individuals. And so remember, the definition of a population is just a group of individuals of the same species that live in the same area and they can interbreed with one another. And what you can get with your populations is sometimes you can get geographic isolation and separation of those populations where those populations don't exchange genetic material. And if this isolation occurs long enough, it can lead to different alleles and selective pressures between those populations, and it may end up in speciation. So an example are these alpine lakes that are the individuals that live in these lakes are isolated from one another, even though they're close by. Only time they would interact is if these ever overflowed and species move through, but that's rare events as well. And so you can get differences in genes and a lack of gene flow here that really affects the gene pool in that population. And so this brings us to the idea of what exactly is a gene pool. And so at a population level, biologists will look at the gene pool of that population. And the gene pool is basically all of the alleles that is expressed or carried by every single member of that population. For a lot of loci, so for a lot of genes, there are 2 or more alleles in that gene pool. Um, for simplicity, we generally talk about only having 2 alleles while teaching courses because it's easier to think about. But in reality, most of the time it's 2, 3, 4, 5, 6 alleles that are present at a particular gene. And remember, each allele is coding for a specific trait, like different color of the eyes, etc. And so, for example, let's look at a wildflower population with 2 varieties of blooms. This wildflower population can have red flowers or it can have white flowers. Red flowers are dominant to white flowers. Dominant alleles are shown by or represented by a capital letter. Recessive alleles are usually represented by a lowercase letter. So in this situation, we're going to say uppercase R is the dominant red allele. Lowercase r is the recessive white allele. Okay. Now let's say that 80% of the population has the dominant R allele. So 80% of the individuals have that dominant R allele. And that percentage is represented by the letter P. Okay. And we can use that to calculate the allele frequency for the lowercase or the recessive R allele, the white allele. We also can use those frequencies to be put into the Hardy-Weinberg formula, and we can calculate the frequencies of the genotypes. Okay. So this is a flower we're looking at, right? So red and white. And these are the formulas that we're going to use. So here we have allele frequencies. You have your P allele, which remember is your dominant allele, so uppercase R. So let's write this here. So uppercase R. Your Q allele is your lowercase r, right? And so we already said that our uppercase, our dominant allele, which is red, is 0.8. Okay. And so frequencies have to equal 1. So you can't have more than 100% in a population. So it has to equal 1. And so what we can do then is to determine the frequency for the lowercase or the recessive allele, which is white flower color, you just say 1 minus P. And so 1 minus 0.8 is 0.2. So the allele frequency for the recessive allele is 0.2. And now you can do the same thing if you knew Q and you didn't know P. So if you know Q but you don't know P, then P would equal 1 minus Q. Right? So you just rearrange this allele formula. Now we can determine the genotype frequencies. So remember we said genotypes are homozygous dominant, which means you have 2 copies of the dominant allele. You have homozygous recessive, which means you have 2 copies of the recessive allele. Then you have the heterozygous genotype. 1 copy of the dominant, 1 copy of the recessive. Okay. We can determine the frequency of these genotypes in the population by using the Hardy-Weinberg formula. So the dominant genotype is the frequency of P squared. Okay, so the frequency of P is 0.8. So you would just say 0.8 squared will give you your homozygous genotype frequency. Frequency of the heterozygote is 2 times the frequency of P times the frequency of Q. And then the frequency of the recessive homozygous genotype, which is homozygous lowercase r, is just Q squared. Q is 0.2. So you just square it. Okay. And so if we look at all this in kind of table form, remember we talked about Punnett squares. So if you look at this, right, the way where this comes from basically is the fact that you get 2 heterozygous individuals to mate. Remember that 1 to 2 to 1 ratio for phenotypes, right? Or I'm sorry, genotypes is your genotypic ratio. So that means you have homozygous dominant, which is RR. So that's 0.8 times 0.8, which is 0.64. Right? That's the frequency for that genotype. Then your heterozygous is 2 of your Q times your P, right? So 0.8 times 0.2 times 2, because each one is 0.16, right? So you have 1 instance of each. So you have 2 instances here. So that's where 2 comes from. So your heterozygous genotype is 0.32. And then your homozygous recessive is 0.2 times 0.2. So 0.4. So basically 0.2 squared. Okay. And that's where the variables come from in your Hardy-Weinberg formula. Now let's say, if you know 2 of these genotype frequencies, then you can always calculate the other one, right? So if we knew P and 2PQ, you would just say 0.64 plus 0.32, right? So that gives you 6, 9, so 0.96. Then you say 1 minus 0.96 will give you 0.4. Right? So remember that has to equal 1 as well. Right? So remember that's equaling 1. Okay. And so the evolution of these allele frequencies is known as microevolution. And microevolution can actually change the gene pool. So Hardy-Weinberg equilibrium basically says that a population is not evolving. So there is no mutations, there's no natural selection, there's no genetic drift, and there's no gene flow. There's no evolution on that population. In order for Hardy-Weinberg to hold true. Now, because we do have generation to generation change in allele frequencies, that generation to generation change of those allele frequencies is what we refer to as microevolution. Macroevolution is like speciation, so the evolution of new species. Micro is, micro is allele frequency change. Macro is speciation, so new species. So macro is basically new species. Right. Whereas micro is change in the allele frequencies. Right. Okay. And so the way that these allele frequencies can change is through three primary methods: natural selection, genetic drift, and gene flow. Natural selection we classify as one of the most important of these processes because it's the only process that really promotes adaptation. So it's the only process that generates the adaptations, the evolutionary adaptations in the population. And we've spent a lot of time already discussing natural selection. Okay? So now what we're going to do is we're going to go into genetic drift and gene flow and actually discuss what these are. So genetic drift is basically a process where you get a change in the gene pool of a small population, and it's primarily due by chance. And genetic drift is stronger on small populations because these chance changes actually lead to significant allele frequency changes. Whereas if you have a large population, that change isn't as effective. Okay. And you can get small populations through a process known as the bottleneck effect. This is where you have a drastic reduction in population size, and you end up with a decrease in genetic variability because you lose alleles from the gene pool and you lose individual variation. If you don't have a lot of variation, then you lose the ability for those individuals to adapt. And so the reason you lose adaptability whenever you lose variation is because, remember, you need variation for selection to happen. And if there's no variation or a lack of variation, then the trait that may be fit may not exist anymore in the population because it was lost during that bottleneck. And so more variation means more of a chance for that population to adapt to changing environmental conditions. So here we have an example of genetic drift. Let's say we have a small population size. We only have 10 plants. And out of these 10 plants, you have these genotypes, right? So you have homozygous dominant for some, one has homozygous recessive, and a couple are heterozygous, okay? The allele frequencies are 0.7 and 0.3. So the uppercase R exists 70% of the time, and the lowercase r exists 30% of the time. So dominant, 70%, recessive, 30%. Now, let's say we have just kind of a random event where only 5% of the plants produce offspring, or 50%, so 5 of the 10 produce offspring, okay? You end up with this frequency. Now you have 50-50, right? Then let's say only 2 leave offspring. Now all of a sudden you're fixed. So fixation. So there's only one allele. That's the uppercase, the dominant allele, 100%. And so just these small random fluctuations can lead to big differences in these small populations. And again, remember, small populations appear one way through bottlenecks. And this is an example of a bottleneck. Let's say you have a population of individuals, they have these alleles. each allele is represented by a specific color. So we have 3 colors in here. And notice the frequency. Lots of purple, lots of green, and only a few orange. Now, say we have a bottleneck happen where only a small percentage of this population survives. And we pour out a few individuals and notice the frequencies now. A majority is purple and only 1 green. We've completely lost the orange. So now the allele frequencies have completely changed in this population to where it used to be probably, half and half with purple and blue with like 2% or so of orange. And now it's mostly 95, 98% purple and like 2% green, which is a big difference from that starting population. And there are lots of species in around the world that have experienced the bottleneck effect. One of those is cheetahs. About 10,000 years ago, during the last ice age, we think they experienced a bottleneck, and that was extirpated even more whenever hunting happened in the 1800s to almost bring them to extinction. And so, because of that reduction in population size, they lost a lot of their variability, and so we're worried now that cheetahs may have a reduced ability to adapt to environmental changes and can be negatively affected by that, and they may potentially go extinct in the future because of that lack of variability if their populations become affected negatively. Another way you can get small population sizes is through a process called the founder effect. The founder effect is whenever you have individuals from a population go out and start a new colony. So basically, let's say we have a population of individuals. A couple of those will go off and start a new population. This reduces the population size to only the number of individuals that were in that population, but it also can reduce the allele frequency. Let's say we have, or the variability. Let's say we have these alleles in the population, allele A, B, C, and D. Okay. Now, let's say that 3 individuals go off and make a new colony, 4 individuals, and those individuals only have the A and the B allele. So all of a sudden, we've lost the C and the D, and that could also affect the frequencies of A and B, depending on how many A's and how many B's were in those individuals whenever they went and founded that new population. And this represents genetic drift, which we'll talk about in a second, in a new colony, but it also explains the high frequency of certain inherited disorders in small human populations, because if a person has that disorder and they found a population, then that disorder is going to propagate through that small population relatively quickly. So another way that alleles and gene pools can change in populations is through gene flow. So gene flow is another source of evolutionary change, and gene flow is where populations exchange alleles with another population or exchange genetic material through reproduction. You can gain alleles or you can lose alleles through this process, and it reduces the genetic differences between these populations. So for example, let's say you have 2 populations. This population has alleles A and B. This population has alleles let's say A and C. Okay, and so these populations are pretty different. They only share 1 allele. They're different by a second allele. But now if an individual moves from this population to this population and mates over here, and they're carrying the B allele, then they can bring the B allele into that population. And so now this population is different because it has the C allele, but it also has all of the alleles as the first population. So the first population is no different in terms of the A and B allele. But then let's say another individual migrates over here and reproduces, and it has the C allele. Now all of a sudden the C allele is introduced to that population, and your genetic differences have been reduced. So now these populations are similar in terms of alleles and well, not allele frequencies, but in terms of allele combinations. Okay, so they both had new alleles introduced to that population. But then on the other hand, maybe there was only like one or two individuals that had the A allele, and those individuals move over, and nobody else in that population has the A allele. They left. So now you lost the A allele. So now all you have is the B and C. And so you can have that instance where you can lose alleles in a population. And so gene flow, you think about like wind pollination, where pollen can drift in the wind and go between populations, or migrating animals moving between populations, and that can lead to new instances, or it can lead to instances of gene flow and connectivity between populations. So last thing we just kind of want to look at is a little, kind of a closer look at natural selection. And so we said that natural selection is really the only one that leads consistently to adaptive evolution, because it's the only process that is based off of fitness values and selective pressures, whereas the other two are random and are acting on alleles that are already present in the population. And while natural selection is acting on alleles in the population, natural selection increases and decreases frequencies of alleles based off of fitness values in those variable allele types. And so let's think about a blue-footed booby who lives in the Galapagos Islands. The bird's body and bill are streamlined, so it can minimize friction and increase aerodynamics as it dives. And it has a large tail that functions as a brake, so it can spread its tail out and push it down to help slow down in the aquatic environment. And then also female boobies tend to prefer males that have the brightest blue feet, right? So here's a booby if you're not familiar. So really bright blue feet, okay? And so females are preferring the males that have those bright feet. And that means that the traits here are going to be selected for through that natural selection, where the ones that are able to move through the water faster can catch prey easier, and the individuals that are mating more often have the brightest blue feet, and so they're going to pass on their offsprings much more often. But one thing we want to mention here too is that oftentimes you'll hear the term survival of the fittest, and we really shouldn't kind of use that, because it is a little misleading, because what we really mean, and so I guess I should say it's misleading if we take it to mean head-to-head competition between individuals, right? Because then you're saying like it's this combat where you're trying to fight to survive. But really, it's kind of this more passive process where it's just determined based on how well you can do in your environment because of the traits that you have available to you. And it's not always this head-to-head competition. And so we can talk about fitness as this relative fitness. So relative fitness is the contribution of an individual to the gene pool of the next generation relative to the others. Okay? And so this brings the question then, what determines the best measure of relative fitness? Pause the video, take a minute, think about this. So, how do we measure relative fitness, and what's the best way? And really it comes down to just the number of fertile offspring that an individual leaves. If you have one organism over its lifetime that has 20 organisms, 20 offspring that are fertile and are going to reproduce, then that means that there are 20 individuals that are surviving from that parent that's passing on its genes. Whereas if you have another individual that only has 5 offspring, then it's not as fit, it's not leaving as much as its genes, and it's not affecting the allele frequency and the allele combinations as much as the organism that's leaving 20 offspring. And so fitness, relative fitness, is really a value or a determination of how many offspring an organism is leaving behind. And so we mentioned sexual selection earlier. And so sexual selection is a form of natural selection where certain traits are more likely to be passed on because they're present in the males and they're selected for by most often females, but it can be vice versa. Females can be choosy too, where males will choose the females based off of traits. And it generally happens in the sexual dimorphic populations where the appearance between males and females are directly associated with reproduction and survival. So you have differences in the way these males and females look, and then, for example, females may choose the brightest colored males, and again, that's determining whether or not those traits will be passed on to the next generation. And so the more common is usually the females selecting their mates and being choosy, but also it could be vice versa where the males are being choosy and selecting specific females. So here's an example of finches expressing sexual dimorphism. So males being differently colored versus the females. And then also you can have competition for mates where maybe the animal that's the strongest with the biggest horns is able to defeat the other male and gain rights to reproduce and have offspring. And there can be three outcomes of natural selection. So that selection process where fitness is determining allele frequency. And if we think about coat color of mice and we graph the color of the population, you'll get a bell-shaped curve where one coat color will be the average kind of most expressed coat color. And then you'll have some coat colors that are on the extreme ends that are some of the more rare coat colors. And if natural selection favors a certain coat color over others, then the populations of mice will change based off of those outcomes. And so there's three general outcomes that can happen. Basically, you can have directional selection, disruptive selection, and stabilizing selection. Directional selection is where you have a shift favoring one phenotype over the other. So let's say we have a mice coat color where you have light white mice, darker mice, and then you have a gradient in between where you have like grayish colorations. Directional selection will favor one of the extremes, so either the completely white or the completely dark color. So the direction will shift to one extreme. Disruptive selection leads to a balance in the extreme phenotype. So it basically removes the intermediate colors. So both the white and the dark color will be favored in disruptive selection. In directional selection, one of the extreme phenotypes, either white or dark, will be favored. In stabilizing selection, this favors the intermediate phenotype. So in our example, it would be the gray phenotype. And so that intermediate will be the most favored, so the curve will stabilize at that intermediate phenotype. And so here's a figure showing this as an example. So we have our original population with the white, the dark, and then the gray gradients in between. For directional selection, we're going to favor one of those extremes. And in this example here, we're favoring the dark color. So the average coat color becomes darker and you lose your white coat colors out of the population. For disruptive selection, you're favoring both of the extremes. So white is advantageous and dark, whereas gray is not advantageous, and so you lose the intermediate color. Stabilizing selection says the opposite. It says gray is advantageous, whereas your extremes aren't, and so you lose your extremes, and we focus on the intermediate, the gray coat color. So now that's more advantageous, and that becomes more more popular in the population. Okay. And so let's think about this question. If you have beak depth in a population of ground finches on an island in the Galapagos, and it increases, so that beak size increases when easy to crack seeds become scarce. This is an example of which mode of natural selection. So is it directional, disruptive, or stabilizing? Okay, so pause the video. Think about that for a second. Okay, so in this example, if you have easy to crack seeds and hard to crack seeds, then the easy to crack seeds, you would have maybe smaller beaks. If you have hard to crack seeds, then maybe you have larger beaks to crack those seeds. And then if you're in between, you only can crack the medium-sized seeds. Okay. If those easy to crack seeds become scarce, then we start to favor the larger beaks that can crack the hard to crack seeds. So this is favoring one extreme, so it would be a form of directional selection. Okay. All right, so that was pretty long, detailed video, but that's it. That's the final video for our evolution module. As always, let me know if you have any questions, any concerns, and we will see you next week in our next module. Take care.